Hi, everyone. Below the screen, my name is Diego Grinman, and I'm a postdoctoral associate in Yale University, where I study the effects of breast cancer in mineral and energy homeostasis. And I'm also a member of the HubLE editorial board, and I'm here on behalf of HubLE to interview Dilara, who presented her work at the ECTS 2021. So Dilara, welcome, and do you want to introduce yourself to the HubLE audience?
Okay, hello, I am Dilara, and I'm a second-year PhD student from Institute of Biomechanics, ETH Zurich, Switzerland. And I'm here to present some part of my PhD thesis that we submitted as an abstract to ECTS. Okay,
great. And what are you presenting actually?
So we generated a fluorescent reporter mouse model for osteoblasts and osteoclasts. And we validated the model with genotypically and phenotypically with immunohistochemistry. So that was the my poster and my abstract.
Nice. So you actually, what are, I mean, specifically the key findings of your work? I mean, there were previous GFP first models that also could see osteoblasts and osteoclasts. It's just something new. I mean, what are your findings?
So we show that our model is a robust model to allowing us to identify osteoblasts in class, and it is done with CRISPR-Cas9, not with the traditional methods like CRE or BAC, and that will allow us to track them, identify them, and also provide us to study their interaction in vivo in their local 3D microenvironment. So this has more advantages than the other methods in terms of specific expression, and it doesn't have any unexpected recombination, and also the toxicity wise.
That's impressive. I mean, it's really useful to have a model like that. And if you can think, in the future, what's the big pictures, the implications of your findings?
Our ultimate goal is to generate prematurely aged fluorescent reporter mice to investigate molecular responses of single osteoblast in class during the bone adaptation and regeneration. And our model will enable us to study age-related osteoporosis by providing identification of the bone cells so that we can observe the changes of the degeneration over age, which will be possible with the model that we will have.
Right, and I guess it's going to be also helpful to isolate those cell types, right? Which is kind of tricky right now.
Yeah, so yeah, because they are fluorescent, we don't need to do another immunohistochemistry to identify them. Under microscope, they will be visible, and it will be easy to identify them, track them, and isolate them, which can have many purposes.
Well, so Dilara, what is next in your career? I mean, you said that you're a PhD student, so are you, thinking about staying in the academia? Are you after finishing your PhD? What are your plans?
I wanted to apply my research skills in applied science. So, I mean, I would like to look for opportunities for working in disease-related systems in maybe biotechnology companies, so that I can transfer my expertise or knowledge about genetically engineered mouse models into products with potential to potentially have positive impact to human health.
Wow.
But it's a long road at the moment. I don't know.
Yeah, well, that's a very, specific future that you have in mind. So Dilara, thank you so much for sharing your work with us. And if you are an investigator, you want to share your ideas on your research, we got your back. HubLE is there for you. So please contact us, and we are going to help as much as we can. Thank you, Dilara, again.
Thank you for having me.